Hello everyone, welcome again to Julsummit Biochemistry Hub. I'm still your host, Dr. Juliet. Today we shall be looking at structures of nucleobases. We must first of all remember that nucleobases are integral parts of the nucleic acids, DNA and RNA, which we know are essential for survival and the transfer of traits from one generation to another. Nucleobases are basically of two types, the purines and the pyrimidines. The pyrimidines have just a single ring, the pyrimidine ring, while the purines have two rings in one, pyrimidine ring and the imidazole ring. If you look at the structures carefully, you will see they are numbered differently. And also, one thing to remember in memorizing the structures is that carbon has a valency of 4, while nitrogen has a valency of 3. So every time we have carbon and nitrogen in a compound, you must make sure the bonds around them are complete. 4 for carbon and 3 for nitrogen. Now looking at this pyrimidine ring, you can see that the ring is a benzene ring, so we see that the double bonds are alternating. And then we see the carbon around this bond around the carbon, we have 1, 2, 3. We introduce a hydrogen to make it 4. For nitrogen, we have 1, 2, 3, it's complete. For this carbon, 1, 2, 3, introduce hydrogen to make it 4. This carbon, 1, 2, 3, introducing hydrogen. This one, nitrogen, 1, 2, 3, is complete. For this carbon, 1, 2, 3, 3, introduce hydrogen to make it 4. And all of that. And also for the purines, the same goes to this ring. Then for this, also you check nitrogen, 1, 2, 3. Carbon, 1, 2, 3, add a hydrogen to make it 4. This nitrogen, 1, 2, introduce a hydrogen atom to make it 4. Now the purine bases are the adenine and the guanine. So you can see they have the basic structure for purines. What's the difference between them? Just as carbon 6. The hydrogen in the parent structure is replaced with an amino group. While for guanine, the hydrogen at carbon 6 in the parent structure is replaced with an oxygen atom, while at carbon 2 is replaced with an amino group. That's the basic difference. So while we have just amino at carbon 6 for adenine, for guanine we have oxygen atom at carbon 6 and amino group at carbon 2. Now, for the pyrimidines, what's the difference between cytosine, uracil, and thymine? We see that basically they all contain the parent structure of pyrimidine. Now the difference is in the substituents at various positions. For the cytosine, we see oxygen atom replacing hydrogen at carbon 2 and amino replacing hydrogen at carbon 4. If you compare guanine and cytosine, you will see that these side groups are the same, but the positions are switched. While we have oxygen up here and amino here, then we have oxygen down here and amino up there. Now for uracil, what's the difference between uracil and cytosine? We see again the oxygen atom remains intact at carbon 2, but now at carbon 4, instead of an amino group, we have an oxygen atom. So we have 2 oxygen atoms attached in uracil, and then we have 1 amino up there and 1 oxygen atom down there for cytosine. What about thymine, the last member of the pyrimidines? Now, thymine is almost the same as uracil, except at carbon 5, where we have a methyl group replacing the hydrogen at carbon 5. So we have oxygen atom up here and down here, just like uracil, but now introducing a methyl group in place of hydrogen at carbon 5. Differentiates uracil from thymine. Now remember that cytosine and uracil are found in RNA, while cytosine and thymine are found in DNA. Now when it comes to the formation of nucleosides from bases, what is nucleoside? Adding sugar to any of these, they become nucleosides. For the purine bases, when they become nucleosides, they end in ocene. So adenine becomes adenosine and guanine becomes guanosine. While for the pyrimidine bases, when they become nucleosides, their names end in idine. So cytosine becomes cytidine, uracil becomes uridine, and thymine becomes thymidine. Now remember, in the formation of nucleosides, the pyrimidines attach to the sugar, ribose sugar at nitrogen 1, while the purines attach to the ribose sugar at nitrogen 9. I trust that we've enjoyed our time together learning structures of nucleobases today. You kindly follow us on Facebook, TikTok, and YouTube to keep updated on our latest tutorials on biochemistry. Thank you so much for your time today. Till I see you next time. Bye-bye.